Translational research. Is translational a dirty word, a black hole, or an opportunity? And I hope that after we've had a chance to talk about it, you'll start to think of it more as an opportunity. But in order to understand what translational research is, we really have to start with what do we mean by basic research. And the National Science Foundation in 1945, I think, did a good job of defining what basic research is. Basic research is performed without thought of practical ends. It results in general knowledge and an understanding of nature and its laws. This general knowledge provides the means of answering a large number of important practical problems, though it may not give a complete specific answer to any one of them. And if you think about the research that is done at Salk, most of the research here is basic research. It's performed not with thought of practical results, but it's thought in terms of scientific results, knowledge results. And of course, that's the mainstay of most academic institutions and their research. Well, what is translational research? Well, I have to say, if you look online and try to find a definition for translational research that makes any sense at all, I wish you luck, because it's like the five blind men and the elephant. Depending on where you're starting from is what you decide that translational research means. So, for example, if you look at what people say, if people are public health officials, they'll say, well, translational research is when you take a look at drugs that are being marketed and you look at the population demographics and the impact on the disease state, and then you take that information to go back and refine the product and its distribution plan. Well, that's not what we think of as translational research. If you ask a clinical researcher, well, they say translational research is doing human clinical trials. And yes, that may be translational research, but that's not at the end of things where we start. We really have to look at translational research starting from the basic research and saying, where do you go next? And a lot of these definitions really don't help. And many of them are multi-part, and there's phase one, translational research, and phase two, and phase three, and it really doesn't help much. So I think that, we really need to understand what we mean by it. I have to say, translational research, the concept came along not that long ago, and I think it's really kind of a politically correct term for what industry has always called applied research. It's research with practical goals in mind. And, if we go back to what NSF talked about in 1945, they said basic research provides a means of answering a large number of important practical problems, may not give us a complete specific answer to any one of them, and that the function of applied research is to provide such complete answers. And I think that's really what translational research is. It's research that starts from the basic research and goes further, but is now focusing on the practical problems, looking for specific answers to practical problems. Now, Francis Collins is the director of NIH. Wrote an article recently in July in Science Translational Medicine, the online journal, in which he talked about the initiatives that NIH now has in translational research in medicine. And I think that his discussion of translational research was a bit more helpful. So he talks about there being 8 major challenges in translational research. The first being discovering the basic pathogenesis of information, assessing if there's a clinical potential, developing diagnostics, therapeutics, and devices, preclinical optimization, regulatory assessment, testing in humans, application for approval by the FDA for widespread human use, and then assessment of how that approved use has worked out in the real world. Well, let's take a closer look at these things that Francis Collins says are really the key steps here. Now, the first one he points to is discovering basic information about pathogenesis of disease. Now this is really basic research because you're really, at least as we practice it here at the Salk Institute, and I don't think there's any doubt that the Salk Institute does this particular step as well or better than any institution in the world. What about step number 2? Step number 2 is assessing if the information has clinical potential. Well, what this means is, assessment is essentially just taking the basic science information that you've gained and thinking about it in a different way. Thinking about it in terms of its practical application. Okay, I've studied this receptor until I'm blue in the face. I now understand it. What can I do with that? How can I use that to treat disease? What impact can I have on a patient? I know that this molecule is involved in glucose metabolism. Is there something I can do with what I know about this receptor that can help me to conjure up in my mind a treatment for diabetes? So it really comes down to starting at some point to think about patients. We tend to think in research, we tend to think about disease in kind of a big way. We think of it as, diabetes, we think about the huge magnitude of the diabetes problem as it's growing around the world. But I think that to assess the clinical potential of what you're doing, I think that one of the most powerful ways to do that is to start thinking about patients. Think about not populations of disease individuals, but think about real people and the challenges that these diseases pose for their everyday lives, and say, is there something I can do with what I know that can have an impact on real people's lives? And it's a matter of, asking questions about what you already know about your basic research. Say, okay, I found this enzyme. Is there something I can do to modulate the expression or the activity of this enzyme that might be able to treat or prevent the disease I've been studying? Or is there something upstream or downstream of there where I could have an impact? And then the question is, what can I do to confirm that? Are there some compounds that I could test? If I think that I could inhibit this enzyme, what kinds of compounds might inhibit this enzyme? Well, maybe I can find that there are some known inhibitors. Maybe they're not very specific, but it gives me a place to start to test whether there is a possibility to develop a therapy based on what I know about this. In other words, you're thinking about what kinds of products might be developed. And new medical therapies always involve products of some kind. They involve new drugs, new vaccines. Sometimes they involve already known drugs, new uses for drugs that are already known. Medical devices, systems for delivering drugs, materials that are needed to carry out new procedures. So if we're thinking about stem cell therapy, you've got to think about, well, how would that be done? I mean, you would isolate somebody's cells. And what conditions would you need to do that? And then you would need to isolate the stem cells. You would have to have the materials in a hospital setting that could do that, and then you'd have to propagate those, and then you'd have to administer them back to the patient. How would that be done? You need to really think about what are the things that would have to be done in the hospital, and what are the products that would be needed to carry that out. And then one of the things that you're often dealing with in basic research is assay systems. And the question is, well, can you take those assays and make them into diagnostic tests, prognostic tests, or ways of testing the efficacy of drugs that you might try to discover, or others might try to discover. Now, as you're thinking about what these products might be, you have to keep in mind that getting FDA approval for a new drug, medical device, or even a diagnostic product could cost well over $100 million. Obviously, you don't have $100 million laying around that you can do that yourself. How do you find $100 million? How do you get somebody to spend $100 million on your idea? And the only way you're going to get somebody to spend $100 million is if they think that by spending $100 million, they're going to get $500 million back. And, companies that big pharma, that's the way they think. You talk to people from Glaxo, they say, well, we don't invest in any kinds of technologies if they're not at least $1 billion a year in sales projected sales. So profit motives is what drive companies and their stockholders to invest in developing new medical devices, new drugs, new medical treatments. It's always about profits because the money, the $100 million, comes from stockholders who've invested in the company, and the stockholders are looking for a return on their investment. Now you say, oh, well, I don't think like that. I'm not a profit motivated person. That's why I'm in academia. My life isn't about money. I don't want my life to be about money. And the nice thing is, when you're thinking about translational research, you don't have to make your life about money. You just have to realize there are other people whose lives are about money. And you have to exploit that. And just say, how can I convince these people that they'll get rich doing this? Because then they'll pick up the ball and they'll run with it with all their money. And, there's a lot of risk. We don't know whether they'll make it to the goal line or not, make a lot of money or not. But if I can convince them they will, they'll put $100 million into what I'm trying to do here and move this technology along. So you don't have to be profit motivated. You just have to realize there are a lot of people who are and do what you can to try to feed that profit motive. Now, the third step in translational research, or the third big challenge that Francis Collin pointed out, is you've got to develop diagnostic tests, therapeutics, drugs, and vaccines, etc., or medical devices. Now, fortunately, when you're thinking about translational research from the Salk perspective, starting from basic science, does not mean that you have to develop commercially viable products. You only need to come up with an invention, a concept for a product that is patentable. So the patent monopoly then can be used as an incentive to get those greedy profit motive people to plunk down their $100 million invested into your idea. And basically what a patent does, if you have a patent, if we get a patent on your idea and we license that to a company, the company knows that if they develop that drug and sell that drug in the marketplace, no other drug company can make that and sell that same drug in competition with them, and they can make a lot of money and they can make enough money to pay them back for their $100 million investment in the product development and make profits for their stockholders. So really, we've talked a lot about intellectual property, but really when it comes to the biopharmaceutical industry in particular, patents are what really are the baton that we can pass from someplace like the Salk Institute to big pharma that will encourage them to make the big investment to get those therapies to market. Now, when you think about what your invention might be, what we might patent, you have to remember that perfection is absolutely not required. You don't have to come up with a commercially viable drug. All you've got to do is prove the concept. So if you've decided that you have this particular enzyme and you think that inhibiting that enzyme can be a useful treatment for diabetes, you only have to show that in a very crude way. Enough to convince the patent examiner that you've demonstrated that, yes, inhibiting that enzyme affects blood glucose. A patent on an invention, the courts have said, this is an old case I cited here, but I think it has a nice summary of the law from 1980. A patent on an invention is valid even if it is imperfect or operates crudely, if it would not be commercially viable as a product, if it doesn't accomplish all of the intended functions, but only some of them, if it doesn't operate under all conditions, or if it is only partially successful. The one case that always comes to my mind was a case between Kodak and Polaroid when Edwin Land had invented the instant camera. And Polaroid made a lot of money on instant cameras, and Kodak tried to move into that business and was sued for infringing the Polaroid patents. And so Kodak attacked the patents and tried to get the court to declare the Polaroid patents on instant cameras invalid. And they put Edwin Land on the witness stand, and they said, the lawyer for Kodak said, "Mr. Land, would you please show us the photograph that was the basis for your patent application on instant photography?" And he held it up, and the lawyer says, "Mr. Land, won't you admit that's just a black square? That's not a photograph." And he said, "Oh no, if you look really closely, you can see the brim of my hat." And the court said, "That's enough." He said, "The fact that you could make out the edge of the hat, that was enough to prove that you could make a photograph." Was it a nice, clear photograph? No. But it did capture a visual image on the film, and that was enough to prove that an image could be laid down. Of course, they perfected it quite a bit after that. So this was a case where it was only partially successful, but that was enough to prove the concept to support the patent application. The fourth step that Francis Collins talks about in translational research is preclinical optimization. Now, this is also something that we can do here at the Salk Institute. Sometimes there are fairly simple experiments that you can do that will make your invention more attractive to investors. So perhaps you've decided that you found a fairly crude inhibitor of your enzyme, and you can show that it does affect blood glucose. Well, maybe you should try varying concentrations of the drug and see if you can show that with increasing dose, you get a bigger impact on glucose levels in the blood. Maybe you could take a look at toxicology a little bit and say, well, how much of this relatively crude drug would you be able to administer before it showed severe toxicity? You might be able to take it and compare it with the blood glucose effects of several commercially available diabetes drugs and show that it works as well or maybe even better under some circumstances in diabetic mice than drugs that are currently on the market. If it's a potential cancer therapy that you have, well, maybe you've been doing your work on prostate cancer, but you might make it more attractive to investors if you test it against other cancer types and find that it has a broader potential. Of course, if all of your experiments up to that point have been done in vitro, if you can put it into animals and show a beneficial effect, show that it actually works in vivo, that can make a big difference for a potential company that might want to develop this into a product. Now, then Francis Collins talks about the last four challenges of translational research, and these things are these are the parts that are very expensive. A regulatory assessment, getting into human trials, FDA approval, and then assessing the impact in real-world use. These things, these are really the things that we expect the pharmaceutical industry to do. So we can work on the first four steps. The first one basically being basic research, but the next three steps being translational steps, and hopefully generate enough enthusiasm by those profit-minded folks in industry that they'll invest their money in developing your invention into the next great drug product. Now, one of the things about translational research is that, or applied research, is it's often a very different experience. So we start out, if you're doing basic research, you're testing hypotheses, your inquiry is very open-minded, you can change path along the way. The results that you're going to get sometimes are unknown until you do the experiments. Your experiments are really designed on a rational basis. You're looking at the theory, what you know about the theory and designing your experiments, and you really care why things happen the way they do. But in translational or applied research, it can be a very different experience because your goal now is not knowledge, but your goal is a particular product goal. You're trying to develop something that has a practical value, and you know what that practical goal is. And very often when people fund translational research programs, because you know what the goal is, they have some timeline expectations that, by the time the year's over and you finish the project, that you will have achieved that particular goal. And they may have particular milestone events along the way that they'd like to see you accomplish to show that you're making progress towards that goal. When companies fund translational research projects, they're always talking about deliverables. What can you give me that, can you get me to a prototype drug in 6 months? That's a deliverable. Send it to us and we're going to test it. A lot of the research in translational research can be empirical, where you don't really care why it's working. You just want to find something that does work. And I'll give you a good example of my own experience with that. So I was working after I finished grad school, I did a postdoc, and I went to work for a biotech company. And one of the first things they asked me to do was glucose 6-phosphate dehydrogenase is an important enzyme used in blood test reagents, for clinical blood testing. And our company manufactured this, and they said, well, they wanted to improve the shelf life of the product because it was sold as a lyophilized powder in a little vial, and they kept a fair amount in inventory, and they wanted to make it so that they could make the inventory last a little longer, and they wanted to know what we could do to stabilize the enzyme so that it would last longer in storage. Well, I was really fresh out of academia. My approach was, okay, well, I'm going down to the library and I'm going to study, what are the factors that disturb secondary, tertiary structures and enzymes, and what kinds of things stabilize those structures, and take it from really a theoretical point of view. Now, the other guy who worked in our lab, he'd been with the company for a few years, and he kind of chuckled at me. He said, well, Bob, he says, you may come up with the best idea. He says, but what I'm going to do is I'm going to go into that chemical closet over there. I'm going to take out 100 vials. I'm going to make up 1% solution of each of those 100 chemicals. I'm going to take 100 microliters. I'm going to put it into 10 vials of the product. So I'm going to wind up with 1,000 vials. And I'm going to put them in a 40-degree oven to simulate, kind of accelerate deterioration of the material to simulate long-term storage. And every couple days I'm going to take out a set of 100. I'm going to run them through the autoanalyzer. And I'm going to plot the enzyme activity as a function of time at 40 degrees in the presence of these 100 things. And I said, oh, who knows what you're going to, why would you think that would work? You just pick these random things out of the cabinet. So it was an interesting test. 2 weeks later, guess what? I'm still reading. Okay? He's already found 3 things that work, and they were fairly cheap chemicals that could be very easily added to the product at a very low cost and would extend the shelf life of that product by 10% in 2 weeks. The moral of the story is, when practical results are what you really need, theory may not be the best starting point. Sometimes, if you just give up on the question of why and you say, what might work, and start testing things, you might get there a little faster. Now, NIH has become very bullish on translational research. Yes?
Curiosity, didn't know if anybody after the fact went to see the how of how or why those 3 worked, or it just was really inconsequential because
really didn't care. And that's what, academic people usually get very frustrated with companies. It's like, well, you found the answer. Well, how come it works? Oh, we don't care about it. We're not spending money to figure that out. We're moving on to the next project. And yeah, it's a little frustrating, but no, they didn't care why. As I recall, one of them was an amino acid. And so, it may have simply been that, if you flood, if part of the degradation was there was an amino acid that was being oxidized, and you flood the sample with that amino acid, you kind of have a protective effect. But that was all speculation because nobody spent any time testing to see why. In the article that I mentioned by Francis Collins, he announced the creation of the National Center for Advancing Translational Sciences. So now this is part of a long march that NIH has had towards trying to assure that the investment that we make nationally in science ultimately has some benefit for the taxpayers who are footing the bill. And I think that the first big step was the NIH roadmap, which where NIH basically outlined their goals for the future and basically said, we're the National Institutes of Health. We're supposed to be promoting healthcare in the United States, and we want to start to pay attention to the outcomes of that research. And so the NIH roadmap included a number of things that ultimately now have become part of the National Center for Advancing Translational Sciences. This new center is aimed at basically taking some of these translational research funding mechanisms that NIH has been developing over the last few years and putting them all under one umbrella. And the hope is that by doing that, collectively, these programs will not only be able to advance translational research projects, but will also be able to identify bottlenecks in the process of moving from basic science to practical results, and find ways to re-engineer those bottlenecks, reduce them, remove them, or bypass them. And some of the bottlenecks are very specifically addressed by some of the NCATS programs. The NCATS, there are 6 NCATS components, and there may be more over the course of time. The one that has been underway for some time is the CTSA program, Clinical and Translational Science Awards. And what they did was NIH used to fund, I think there were about 120 clinical research programs at American universities. And they did an assessment of them, and they discovered that the vast majority of the clinical research that was being funded by NIH was not leading to products. It was leading to interesting understanding of various drugs, but it was not leading to new therapies. And the reason is that the academic clinical researchers were not really connected to the company side where the products get developed. And so they restructured, they basically said, we're going to end our clinical trials funding program and replace it with the CTSA. And the CTSA program, to apply for it, you have to offer a PhD in clinical and translational research. And you have to teach translational research as a separate discipline, including you've got to teach about regulatory, about FDA approval process. You've got to teach about high throughput screening. You've got to teach about toxicology and all of the kinds of things that are important parts of the process of developing new drugs. You can't just say, gee, let's see if you give this drug what effect it has on disease X. It has to have a practical goal. And it's been very difficult for a lot of institutions to recapture their clinical research programs because there's only 60 CTSAs, and many of them had to apply 2 and 3 times before they finally had focused enough on the translational side that the NIH was willing to grant those programs. The one other I'll point to here for a moment is the NIH-FDA Regulatory Science Initiative. Part of the one of the big bottlenecks in the drug development process is getting drugs through the FDA approval process. And NIH is going to try and work with the FDA to see if they can streamline that. Now, fortunately, we have someone here at Salk who knows a lot more about these programs than I do. Mike Nunn. Is Mike here somewhere? Rachel, have you seen Mike? Okay. Well, Mike Nunn is the executive director of our research development, and I was hoping he was going to come here today and talk to you about some of these other programs. I'll tell you a little bit about the Molecular Libraries program. Is essentially an NIH program aimed at doing translational or doing high throughput screening in an academic setting. And so NIH basically put out a challenge to academic institutions to put together high throughput screening programs similar to what industry has, where you essentially are able to set up assay systems, and using robotics, you're able to test tens of thousands, if not millions, of compounds for their impact on particular disease states using these assays. And there are a lot of academic scientists who come up with assays that are very useful in developing new drugs, screening for new drugs. And so the hope is that by having academic centers involved in the high throughput screening program, that they'll be able to attract more target opportunities and identify early leads that can then be passed off to industry for more refined medicinal chemistry development. And there were a number of programs that got started, and the two that got approved ultimately for a very large amount of funding. One was here at the Burnham Institute, and the other is at Scripps in Florida. And they both have sizable libraries that they got from NIH that they can screen your targets against. And they also have their own private libraries that are substantially larger. I think both programs now can screen over 1 million potential drug candidates against your targets. And well, how do you convince them to screen your target? Well, the NIH has a special grant program where you can apply and you can get funding for your screening program, and then the funding then goes to one of these centers to carry out the screening on your drug. And of course, what NIH wants to know about in your application is they want to know why your particular target is a good target candidate for developing drugs, and what kind of drugs you think you might be developed, and what you think the therapeutic value of being able to interact with that particular metabolic receptor, for example, might have on the disease of interest. And then they also have a program on therapeutics for rare and neglected diseases. And this is closely allied with the FDA's orphan drug program. And the orphan drug program has turned out to be really a vital shortcut to FDA approval for many drugs. The Therapeutics for Rare and Neglected Diseases program is essentially a funding mechanism for those who want to look for drugs that would treat diseases that are fairly uncommon. So there are many diseases that, connective tissue diseases, for example, where there may be only a few thousand people in the world that have those particular diseases. And if you had to go through the typical process of drug development for that Big Pharma does, and then go through the ordinary FDA process, no big company would invest, because there's just no way to make enough money to earn back revenues that would cover your costs, let alone profits for your stockholders. So there are a lot of diseases that simply would never find a cure or a useful treatment. Well, the FDA program on orphan drugs basically says if there is a disease state that has a limited population, that the FDA will actively help you design your clinical trials so that you can use a small population of patients. They take into account the size of the potential market, and they also take into account the seriousness of the disease and allow you a much shorter path to regulatory approval than you can get for other kinds of drugs. When I was at University of Virginia, we had a patent on a potential drug for treating ALS, which is Lou Gehrig's disease, which is basically a fatal degeneration of your nerves. And we licensed it to a company that developed it as an orphan drug, and the FDA was quite helpful to them in allowing them early access to human patients because there really weren't any good animal models. And since ALS is ultimately fatal in every instance anyway, the risk of giving it to end-stage ALS patients was considered relatively minor compared to the potential benefit for all of such patients if they could identify a treatment. So one of the things that companies often do is they'll look for an orphan indication for a particular drug and then also look for a broader indication for that same drug. So, and very often things that are getting developed for orphan drugs today are things that where it might be very helpful for this small population of very sick people, but also might be useful for a broader population. And I think in this case of the ALS drug, there was pretty good evidence that not only would it be useful for treating ALS, but that it would also be useful for treating neuropathic pain, which is a problem, for example, that diabetics get from degeneration of their nerves that results from their poorly controlled insulin levels. So the incentive, the orphan drug program at the FDA gave them a cheaper way to get the drug approved. And then once it was approved and was already being used to treat humans, then going back and saying, okay, FDA, now we want you to approve it for use in treating neuropathic pain, the clinical trial demands to get that secondary approval were much less. So again, that helps to drive people to develop orphan drugs. Let's see. So we never found Mike, did we? Anyway, well, I'm sorry that Mike isn't here because Mike knows a lot more about these programs since he's involved in some of the review programs at NIH that relate to these programs. But anyway, that's about what I have to say about translational research. Oh, I've gone. Does anybody have any questions? Yeah.
This is minor. I believe that your office used to be Office of Technology Management, and now you're Office of Technology Development, implying a more active role in actually getting patents for discoveries made here in the labs and hopefully promoting them to the outside world where there might be funding to get them out into the outside world. Can you talk about that at all?
Yeah, well, actually,
You are and how much, because you still don't have a huge staff
right.
compared to all the scientists here.
When before I got here, it was called the Office of Management, Technology Management and Development. So it went from technology management and development, and management. I don't think the management's the important part. That's kind of like saying we're the Salk Institute for Administration and Research. I mean, it's not the important part. I think that, but I think that your point is well taken, because I think that we do need to go further down the process. And I think that when I look at the challenges that we have, part of it's personnel, and we're actually in the process of hiring 2 more licensing people. Part of it is transactional efficiency. We do a lot of licenses for cell lines and reagents and things like that, and we're trying to consolidate that into working with some commercial vendors like Millipore that distribute reagents a lot more efficiently than we can, and that will help to cut down on the staff time consumed by those things. But I think that we really do need to be much more involved in helping people to envision the product goals from Salk Technologies. And that requires us to actually get out and work with scientists more closely, be more visible in the laboratory so that people know who we are. They can ask us questions when they see us there. Have our office people that actually do come in the lab and talk with people, get to know everybody. And then also, I think that when it comes to evaluating inventions, I think that because of the historical focus the Institute has had on basic research, which we certainly respect, I think our office has to do more in translational thinking than many other tech transfer offices do. And I think that one of the things that I would like to do, and I think it's certainly within our capabilities once we have the people, is when we get an invention disclosure, not only take a look at patentability and think about what the product might be, but really take a closer look at what's the market for that product, what's the population of patients, what are products that are already in the market that are used by that particular population, what are the steps of commercial development that need to be done to get from the invention that we have to the marketplace, and also take a look at what recommendations we might be able to make in terms of proof of concept experiments that might be done to help give us a stronger patent position that would allow us to get industry to be more excited about making investments in those technologies. And I think that those are really things that we can accomplish, and those are the things that are very much on my radar screen. But I think that there are a lot of infrastructure things that we need to do to free up people's time, to have more time to focus more on those kinds of aspects of inventions. We also have been talking with the administration about the idea of putting together a proof of concept fund, so that when we look at these inventions and we decide that there are certain experiments that would really make a big difference in terms of the strength of the patent position we'd be able to get, that we would have access to some funds that we could make available for people to do those experiments, which I think would make a big difference in moving Salk technologies to the marketplace. Yeah.
Does the Salk have any licensing of technologies to any startups at common?
Yeah, we do. There has been a history of licensing inventions to startups. There are a number of startup companies that the Salk faculty have either started or been co-founders of over the years. For a lot of early stage technologies such as come out of Salk, startup companies are an important outlet, because there are a lot of technologies that are just too pie in the sky for big pharma to be willing to invest in. And the whole biotechnology industry has been based on startups largely because of that. It takes a significant amount of vision to see getting the first stem cell technology to clinical marketplace, for example. Now, there are some big companies investing in that, but there are a lot of startups that have a passion for it and are able to attract investors who individually can share in that vision. Very often, startup companies are the only possibility. For example, many of you know about Andy Dillon's research on extending your life term, and mechanisms how that might be done, and perhaps developing drugs to do that. Well, that's the kind of technology is a very tough sell for big pharma. The first thing they say is the insurance companies aren't going to want to pay to make you live longer, because then they have to buy you more drugs and they have to send you to the hospital more often. If you had one that was shortened the lifespan a little bit, they might be more in favor of. So, to convince big pharma to go chasing after the fountain of youth is going to be pretty tough. I mean, you're going to really have to have a compelling case that it can be done. And you're going to have a market plan that's going to explain how you're going to distribute this drug and how you're going to convince the insurance companies to pay for it. So that's the kind of case where, a startup company with the right-minded individuals, and you might find wealthy people, or generally referred to as angel investors, who are willing to invest in that. So, if I was 98 years old and I had a lot of money, I'd say, hey, yeah, let me put some money into that. That sounds like a great idea. Now that's not a corporate point of view. That's a very personal point of view. So that's the way it works. Now, startup companies are very challenging right now, because what happened was, when the stock market took a beating in 2008, there were a lot of biotech venture funds. And if you take, for example, a venture fund that might have $100 million under management, well, they don't actually have the $100 million. So what they do is they have a bunch of investors, each of whom put in $2 to $5 million. And when the venture fund decides they're going to invest in a company, they put out what's called a capital call. And they ask all of the investors to send in a check for their share of the $10 million they're going to invest in this new company. Well, after the crash, when venture funds put out a capital call, they got so many of their investors said, hey, I got wiped out. I'm out. I'm quitting the fund. So now they thought they had a $100 million fund. Now they got a $30 million fund. And now they got to go back and ask those investors to put in 3 times as much to fund this company. And now they get more people dropping out. So what happened was, after this happened to a few venture funds, all of the venture funds said, no more capital calls until the market comes back. Well, when they said no more capital calls till the market comes back, the only ones they were willing to do is if they've already invested $10 million in a company and that company is about to go bankrupt, they'll give them a little money to survive. But they've been avoiding new investments like the plague. Well, it makes it pretty hard to start a company if the venture funds aren't investing. So you have this large investment pool, often referred to as an overhang of money that should be available, but the fund managers are afraid to go ask for the capital. And it's had really a devastating impact on biotech startups. Lots of places, including San Diego. I mean, it's really taken a big dent out of startups. Now, one of the things that happened when this all started to come about, Big Pharma said, wow, look at the bargains. Because we got all these biotech companies who can't get money from their venture fund partners anymore. And they've got 2 choices. They can either sell the company to us for 10 cents on the dollar, or they can go out of business. Guess what? They're going to sell us the company. So Big Pharma went out and bought up all the biotech companies that they thought were any good. I was talking with Reed Adler, who's he's the guy who from Merck who puts research funds into academic institutions. And he was saying, well, what happened was Big Pharma went through and they bought up all the good biotech companies. Well, Big Pharma used to contract with the biotech companies to do certain kinds of research. But all the good companies they used to contract with are now owned by their competitors, so they can't contract with them anymore. And the only biotech companies that are left are the ones that Big Pharma doesn't like. And so they said, well, now who are we going to contract with to do this kind of research that we used to get the biotech industry to do? And I say, well, we're going to have to go to research institutes and universities because they can do the same kind of stuff. And so that's what's now driving the move of industry money into funding academic laboratories. So in a way, you know, what's been bad for the biotech industry has been good for academia. And you're seeing some unusual structures being put together where they're really trying to treat academia as a subcontractor for projects that industry is working on. And one of the things that we're seeing is a common mechanism developing is where Big Pharma says, well, we'll come up with drug candidates, but we'll let academia do the biology part because they do biology really well. And so we'll give them drug compounds to test, and they'll test them and give us back the results. We'll have the patents on the drug compounds. But if the drug turns out to be successful, we'll make certain milestone payments to academia, and of course we'll pay for the research that we're asking them to do. And so you're starting to see, I guess Pfizer's been the biggest mover in that direction. Pfizer now has a series of big centers. They started one in, I think they've got one in Boston. I think they've got one, Paul, where are the other ones? They've got one in UCFS and UCSD, where they basically say, we're going to set up a company laboratory that's going to do chemistry, high throughput screening, and stuff like that. And they're going to have close collaborations with not only the basic scientists, but also the clinical researchers at these academic institutions to help us to develop drugs. The other things that's going on in Big Pharma today is when the stock market took such a big tumble, their profits were being squeezed. And another reason they're moving more towards funding academia is they basically gutted their R&D programs. You know, they laid off a lot of their scientists. Now they got rid of basic research long ago. Used to be Roche had a molecular biology institute that was really basic science, and those kinds of things went by the boards years ago. But now they're not even internally doing a lot of product development other than chemistry, other than medicinal chemistry. The other thing that's happening is that about 80% of all prescription drugs will have their patents expire by the end of 2012. So big money makers like Lipitor are going off patent. And companies that have had huge revenues on these, all of a sudden the generics are going to be in there and the market value of that drug is going to drop like a rock. And so you've got Big Pharma looking down the road at that and saying, well, how are we going to replace that? We don't have any new patented drugs that are going to replace those revenues in the near term. So Big Pharma is contracting a lot, and they're looking for more economical ways to continue their products development cycles. But some companies like Merck got caught completely flat-footed. I mean, they just didn't have anything in their pipeline that could make up for some of the expiring patents. And this worry about the pipeline drying up was what was driving them to buy the biotech companies when biotech companies started to get wobbly in terms of financing. So we've had really a big consolidation and change in pharma over the last 2 or 3 years that we're still waiting to see where that's going to all shake out at the end of 2012 when all the big money goes away. Yeah.
Just a small point you mentioned before about how Big Pharma wouldn't look at you unless you had $1 billion, et cetera, market cap. But I think precisely because they're really worried about the pipeline and all of the generics coming online very soon when the patents go away, that interested in looking at niche products, and so that maybe some of the biotechs before that your market cap is just too low, they're sending out feelers and
Yeah.
so there is hope if people have startups that are slightly smaller.
Oh, yeah, I think you're right. They're willing to take things in earlier stages of development, willing to take things that have more modest profit potential. And one of the other things that we're starting to see in the inquiries that we get, there's a sudden interest in diagnostics and biomarkers that we haven't seen in a long time. Used to be back in the 80s when I was running the tech transfer program at Rutgers, diagnostics were hot. Everybody was looking for a new diagnostic because if you came up with a new diagnostic test, every doctor would prescribe it whenever they sent you for blood work. Well, that heyday died when Medicaid came out with new rules on, they called the DRGs, the Diagnostics Related Group. They said, if you have anemia, we're only going to test your iron. We're not going to allow them to test for Tay-Sachs disease and every other damn test they've got out there. We're only going to test it. If you have a broken foot, we're not doing blood work on you. So we'll do an X-ray, but we're not going to do the battery of blood work. So all of a sudden, what used to be kind of an open field of making money on diagnostics, the door slammed shut. Interest in diagnostics really dropped dramatically over those years. Well, what's happening now is, the thing is, it's a lot easier to get a diagnostic product approved by the FDA. So if you're a startup company today in the biotech area, investors are scared to death of the 10, 20-year lead time to a product. And so now what people are saying is, well, okay, so we'll have a therapeutic product that we're working on, but let's also have a diagnostic product, because the diagnostic product, we can actually plan to sell some product in 5 years compared to 10 or 15 years with our therapeutic. And it gives us something for our investors to be enthusiastic about in a more realistic time frame. And one of the most interesting aspects of that is they're looking at diagnostics not in kind of a new way. They're really thinking ahead towards personalized medicine. So we had an inquiry not too long ago from a company that said, well, there are a number of drug companies that are developing cancer drugs of a particular class. And we'd like to license this diagnostic assay that Salk has, because we want to develop it into a diagnostic product that you could use on a particular patient to determine which of the drugs of that class might work on that particular patient. Now, the companies that are developing those drugs, they don't want to have such a test, because what they want is, they come up with a new cancer drug, they want every doctor to give it to every cancer patient of any kind, right? And they don't want somebody to say, oh, if you do this diagnostic test, you'll learn that that particular drug only works on 1 out of 5 patients, and you know which patients those are. Now, you would think for practicing good medicine, they'd be smart to do, right? But big pharma sometimes thinks more about making money than they think about making people well. So now we've got other companies that are coming along and saying, well, we're going to come up with the personalized medicine diagnostics so that, and once we come up with it and we take it to the FDA is going to go, oh, well, this is great. We're going to make all these drug companies use that test in the clinic, and then Medicaid and insurance companies are going to say, you've got to do that diagnostic test before you select which cancer drug you're going to give to this patient, which I think is going to improve healthcare, quite a bit. I was when I was at UVA, I was talking about personalized medicine with a neurosurgeon that primarily treated people who had neuroblastoma, which is an always fatal brain tumor. It's a very aggressive and rapidly growing tumor. And he lamented that the problem was that there are about 6 or 7 drugs that you can use to treat glioblastoma. He says the problem is the patient only lives long enough to try 2 of them. And he said, he was desperate for finding a way to figure out which were the 2 to test on that patient. And he was trying to develop some screening technologies where you could take tissue samples and you could determine what the impact of particular drugs on that disease might be. So I think that in a way, the changes in the market driving people towards diagnostics may actually end up being a stimulus for the development of personalized medicine that I think Big Pharma otherwise might kind of drag their feet on. Any other questions? Yes.
I hope this is not something I'm asking about your political position, but I'm curious about the Obama solar battery issue, like the recent one, $500 million funding for the solar battery.
Oh, that.
I didn't follow up really the details, but I was curious to know if you follow some basic research or translational research, or how do you see it?
Well, yeah. I don't know too much. I mean, I've heard about it. I mean, I know people are trying to make it into a political issue, but the federal government invested $500,000 or $500 million in a solar panel company, and for whatever reason, they didn't realize that this solar panel company was costing them $6 to make solar panels, and they couldn't get anybody to pay more than $3 to buy them, and their competitors were selling comparable solar panels for $1. So needless to say, they went bankrupt, and now there are being some questions asked about why in the world they invested so much of our tax dollars in that company. Why didn't somebody take a closer look at the business model? I don't know. I think that figuring out what companies to invest in at the early stage, it's a roll of the dice, no matter what the company is. I mean, I think that a lot of stimulus money was put into alternative energy, I think for very honorable reasons. I think the big question about that particular investment is that President Obama used to own some stock in the company that was the predecessor to that company, and some people who raised money for him were executives in that company. I think that's really what that's about. But there's a lot of, it's always a big challenge about where the federal government invests their dollars into research, and needless to say, we'd like to see them spend more money on basic sciences, things like neuroscience and stem cell biology and things like that. But every administration has its own focus. There was a time, I can't remember when it started, whether it started in the Clinton administration, but there was a period of years where the goal was to double the NIH budget, and that was accomplished. But unfortunately, they kind of ended there, and the NIH budget hasn't really grown substantially since then. It's contracted a little bit. So I have a friend who was director of licensing at the National Renewable Energy Research Center in Colorado, and he said when the stimulus came down, their research budget doubled in 1 year from the stimulus money. That money's been spent, and now Congress is talking about budget cuts, and now everybody who works there is worried about whether they're going to get laid off. So it's a crude process when the marketplace develops technologies. It can be a little bit more erratic when government gets involved because they may not be as sensitive to the long-term commitments that are needed just for some of these technologies. But it's the political realm, and it's the silly season of leading up to the election. We're going to hear all kinds of crazy things. I don't know. My particular spin on politics is I see it as a team sport. and there was actually a study done with functional MRI not long ago. And they took some avid sports fans and they showed them pictures of, of people from the team that they love, and showed them pictures from their rival team, and looked at the way their brains lit up. And then they took some very strongly political people and they put them in the functional MRI, and they showed them some of their favorite, people from their party and people from the other party, and the exact same parts of their brain lit up. So, which makes perfect sense to me. I mean, I think it really is a team sport, and you pick one team or the other, and you root for them. And everybody has their own particular blend of criteria for how they decide which team they're going to be on. Yeah.
On the new changes to the patent system to first to file, does that?
Oh, yeah, I'm glad you asked that. Yeah. Well, there are some really very interesting changes. The bill is like 88 pages long, and there's a whole bunch of changes. But the one that most people are familiar with is it used to be, and really for historical reasons, that in the United States, whoever thought up an invention first had the right to the patent, even if somebody else came up with the same idea later and got to the patent office first. And it was really a big cornerstone of our system of Yankee ingenuity, and it was a major battleground, particularly during the Industrial Revolution, when people were coming up with great inventions left and right. And what happened, the process that you went through is if you filed a patent application and the patent examiner sees that there's another patent application on the very same invention, they would declare an interference, which is a kind of a legal contest in the patent office to decide who thought of it first and was diligent from the moment of conception all the way through when they reduced the invention to practice and filed their patent application. And it could be expensive, it was tedious, and the problem is the rest of the world didn't do it that way. So when you file a patent application in Europe or in Japan or anywhere else, whoever files the first patent application has the right to the patent. They don't have this interference thing. Now, there have been patent harmonization talks going on internationally for at least 35 years to try and, because the cost of patents is hideous. To get a U.S. patent, if you're lucky, it'll cost you $20,000. To get the same patents on the same invention in all of the economically significant countries in the world is probably going to cost you another $120,000 for a single invention. So there have been efforts to try and harmonize the system, and we have a number of things that have helped a lot. So it used to be you had to file in each individual country in Europe. Now they have the European Patent Office. You file a single application. When that patent gets approved by the EPO, then you can certify it in each one of the countries. So you don't have to go through 27 patent offices. There's also something called the Patent Cooperation Treaty. So you can file a single patent application in the US, and you can turn that into a patent application anywhere in the world within a year. So by filing one application, you preserve your rights around, you have 30 months to decide which countries you want to continue in. So that helps. But the big barricade to patent harmonization has been this U.S. first to invent system. And so we could never have a world patent because nobody else would agree to this first to invent system. So it's really a step towards patent harmonization. One of the other driving forces for patent harmonization was the World Trade Organization. So a number of years ago, in an effort to eliminate trade barriers, much like NAFTA did in North America, we in the United States and the other major economic countries formed the World Trade Organization. And one of the requirements was that you modify your patent laws to make them stronger, at least a certain level of reliability, and that they also had to have certain features. And again, the effort is to try and harmonize the way patents are handled around the world in all of the major economic trading countries. And they wanted us to go to first to file back when we made changes to our laws for that, but that was in 1995, and you just couldn't get things started. So the big difference in the first to file system for us, it's not really that big of a deal because we have to worry about racing to the patent office anyway, because with a new drug, you need coverage in all of the major countries. So you can't rely on the first to invent system in the US. You'll lose your rights in Europe, and the value of your technology is shot. So now in Europe, if you go give a seminar, it's whatever you say is no longer patentable, and that's never been the case in the United States. Not clear to me from the wording of the Patent Reform Act whether that is now an issue for us. But basically, whoever gets to the patent office first gets the patent, unless there's provisions that allow you to prove that somebody stole the idea from you. And there is a grace period. So if you go give a talk, you have up to a year after you publish a paper. You have up to a year after you publish your paper in which to file your patent application, and your patent application will still be good in the U.S. If you publish first before you file, you still don't get any rights in Europe, but you'll still get your patent in the U.S. If you give a talk, let's say you tell somebody, you give a lab seminar. Somebody in your lab tells somebody over at UCSD, UCSD files a patent application on your invention. You file a patent application, and the patent office says, sorry, it's not patentable because UCSD filed this application. You can initiate a derivation proceeding to prove that somehow they got the idea from you and that the patent actually should belong to you. So it kind of replaced one complicated system with another complicated system, but the ultimate result is that first to file is now the rule around the world, and perhaps eventually we'll wind up with a world patent. There's also something called the Patent Prosecution Highway, which is an experiment the Patent Office is doing to try and coordinate the examination of patents between the U.S. and other countries so that it's cheaper and faster to get patents approved around the world. So I think there are a number of other efforts that will come along. There are a lot of other changes in the law. A lot of them are very picayune procedural changes that will just make the process faster and simpler. There's a whole bunch of new fees that you have to pay to the patent office to get a patent. It used to be you filed a filing fee. Now you have to pay a search fee, and you have to pay an examining fee. And so the hope is to get more money into the patent office so that they can hire more examiners, because right now in biotechnology, if you file a patent application today, it will take 3 years before a patent examiner even reads it. And it'll probably take you 5 years to get your patent. Well, I mean, that's a long time to have to wait. And in the meantime, people are investing money. You're licensing it to a company who doesn't know if they're going to get the patent or not, or how broad the patent will be, because the process is so slow, and so it makes people reluctant to make the investment needed in developing new products. So they're trying to whittle down the backlog, and part of the problem was that Congress, for many years, has had a practice of taking money from the patent office. So the patent office would collect all these fees, and they would take a certain percentage of it and they would dump it into the general fund and use it to fund God knows what, pork barrel projects and things. And patent attorneys across the country have been complaining about that for years. They say, we've got this big backlog, and here Congress is stealing money from the patent office. You know, these are fees, user fees we're paying. It ought to go to paying to get patents. And so one of the other things in the bill basically sets up a permanent fund for the patent office and says whatever money they collect will go into that fund and can only be used for patenting. One other interesting thing that's in there is provision allowing the patent office to set up regional offices. Right now, there's just the U.S. Patent and Trademark Office it's actually in Alexandria, Virginia. And the patent office has, for many years, been doing what they call hoteling. So patent examiners don't all have offices. Most of them work at home today. But they have to come into the office twice a month for meetings with the boss and training programs, and also to have interviews with patent applicants. Well, there are some people who live in Chicago, and they fly to Washington D.C. twice a month to go to the patent office. Well, some people will do that, most people won't. And so it means that the technical expertise that the patent office gets to call upon are people who live in the Washington, D.C. area. Yet you would think that for prosecuting patent applications, say in electronics, it'd be great to have an office in Palo Alto, Silicon Valley, where there's lots of highly trained people, and you could hire some great examiners there. So the plan is to set up regional offices so that instead of having to go to Washington, D.C., they could go to the office in Palo Alto. And the first regional office is going to be in Detroit. And you would say, why Detroit? Well, unemployment's big in Detroit, so maybe there's a lot of people they can hire. But Detroit, of course, is the center of the auto industry, and the auto industry has lots and lots of patents on mechanical stuff, and there are a lot of people who have expertise there. And it's going to be the Elijah J. McCoy Patent Office. Anybody know who Elijah J. McCoy is? What's that?
The guy who was our trustee.
Who was what?
Who was our trustee.
No. Elijah J. McCoy was an inventor back during the Industrial Revolution. And Booker T. Washington in 1909 heralded him and said that Elijah McCoy was the most prolific black inventor in history. And mostly what he invented was automatic lubricating systems for steam engines. And he was especially known for one of his inventions, which was a little cup that would automatically lubricate the gears on the machine engine, or on the locomotives. And locomotive engineers, when they would examine their locomotive, they would look at that little cup and they would say, well, now, is that the real McCoy? And now you know who Elijah J. McCoy is. Thank you very much. Let's go have some lunch.